Hey guys, it's Mrs. McQuaid again, and today we're going to be discussing the scientific method and the importance of peer review. If you want to print off slides that you could take notes on them, you can visit my website at www.sevengablescience.com, then select AP Biology Unit 1. The objectives for this unit are to be able to identify components of the scientific method and design controlled experiments, to be able to differentiate between a scientific theory and a hypothesis, and to be able to differentiate between authentic peer-reviewed science in respected journals and pseudoscience in predatory journals. Most of the vocab from this section, just like last lecture, should be a review from previous science classes, but I recommend writing these words down in your notebook, and as you see them come up, copying down a definition for each of them. All right, let's get started. So science is a way of knowing. It's really an approach to understanding the natural world. At the heart of science is inquiry, which is a search for information and explanations of natural phenomenon. And while there are many different ways to pursue science, there are certain characteristics that the scientific inquiry process always uses, like making observations and forming logical hypotheses, and then, of course, testing them. Most scientific inquiry begins with observation. Observations can reveal important data about the natural world, and data falls into two big categories, qualitative and quantitative. Qualitative data are going to be descriptions, not measurements. For example, Jane Goodall is famous for her observations of chimpanzee behavior. Quantitative data, these are going to be things that you measure and record. Oftentimes you'll find them organized into tables and graphs. Collecting and organizing data can lead to important conclusions based on a type of logic called inductive reasoning. And in inductive reasoning, we derive generalizations from large numbers of specific observations. So, for example, we now know that all organisms are made of cells, and that was a conclusion that was reached after several scientists looked at many different types of organisms under a microscope, and they all saw cells over and over again. Quantitative data in science is going to be measured in SI base units. So length would be measured in meter, mass in grams, time in seconds. And those are the ones that most likely you're going to see in this class. When you take chemistry, you'll talk about moles to measure amount of substance. You could see temperature in this class as well, measured in Kelvin. And there are prefixes that go with those as well. So the SI system is based on multiples of 10, and we can use prefixes to indicate like which multiple of 10. So for example, the prefix kilo means 1,000. So if you have a kilogram, you have 1,000 grams. Milli means 1/100th. So if you have a milligram, that's 1/100th of a gram. And in classes like this, you will often work with things as small as the micro level. So micrograms or microliters are common units of measurement. So our innate curiosity often stimulates us to pose questions about the natural basis of phenomenon. So we want to know like how or why something happened. A hypothesis is an explanation based on observations and assumptions that leads to a testable prediction. So think of it as an explanation on trial. A scientific hypothesis always has to lead to predictions that can be tested through experimentation. For example, you might make the observation that your flashlight doesn't work, which of course leads to a question, why? So there's a couple different hypotheses you could explore. The first one is maybe the batteries are dead. The second one could be that your light bulb is burnt out. Both of those are testable hypotheses. A type of logic called deductive reasoning is built into the use of a hypothesis, while induction entails reasoning from a set of specific observations to reach a general conclusion. Deductive reasoning involves logic that flows in the opposite direction, from the general to the specific. So from a general premises, we can extrapolate to a set of specific results that we should expect if the premises are true. And we can then test the hypothesis to see if the results were as we predicted. It's important to note that you can always come up with a different hypothesis for a set of explanations. So, for example, if we go back to that flashlight thing, you replace your flashlight light bulb and it works. And while that does support the fact that your bulb was burnt out, it also could be true that your first bulb maybe just wasn't installed correctly. So there's always different hypotheses that you could test. All right, so a hypothesis we know has to be testable, but it also has to be falsifiable. It's worth noting that we can never prove a hypothesis is true. We can only fail to falsify a hypothesis. And while we can't prove a hypothesis, we can test it in various ways by lots of different people with lots of different experiments, and we can greatly increase our confidence in its validity. So in science, you're not going to hear people say, we proved this. You're going to hear people say, or at least a good scientist will say, the evidence supports this. It's also worth noting that science only deals with the natural world, right? So you can't come up with a hypothesis about anything like supernatural or religious, right? We can't test whether ghosts exist, right? If you can't see it, measure it, and manipulate it, it's out of the realm of science. All right, so when we think about the way that researchers answer questions about the natural and physical world, we often picture this kind of idealized scientific method, right? And this should look familiar from freshman biology and chemistry and all of the other science classes you've taken, right? So it starts by asking a question, doing background research, constructing a hypothesis, you test it with an experiment, analyze data, and then draw conclusions. But in the real world, scientific endeavors never kind of adhere rigidly to the sequencing of these steps. Oftentimes, they can happen out of order. So, for example, in what we call cookbook science, which are the labs that we often have you do as high school students, there's a very controlled design and you follow the steps and you get a result, right? But in real science, we don't know what the result is going to be, and oftentimes we can have like procedural problems, right? And so it's not straight ask a question all the way down. Sometimes your procedure isn't working and then you end up having to spend 6 months troubleshooting that. Sometimes you get partial results, right? And so it seems like maybe your hypothesis is kind of supported, but not really. And then you need to go back to use that experimental data that you've collected and then kind of go back to your original hypothesis and reevaluate it and see if there's a new way to study it or change it. And so these steps can happen where it's almost like a cyclical thing. So doing one experiment often leads to more questions. There's never like a straight up end to science. It's also important to note that there's even non-hypothesis driven science. So hypothesis-driven science kind of goes in the sequence you see here, but non-hypothesis-driven science is going to be something like where a scientist might say, let's sequence all of these genomes or look at gene expression levels for all of this tissue or these groups of people and then compare them to different groups and see if we notice patterns, right? And so when they do that, they're not looking for something specific, they're taking these big generalized patterns and then they zoom in. So really, science can happen in any sequence, up or down, cyclical, whatever. All right. So we know that there's flexibility in the scientific method, but when we carry out an experiment, the researcher has to manipulate a factor in the system and then observe the effect of the change. So in a controlled experiment, that's going to be one that's designed to compare an experimental group to a control group. Both the factor that's manipulated and the factor that's subsequently measured are what we would call experimental variables. So the independent variable is the factor that is manipulated in the experiment, and the dependent variable is the factor being measured that is predicted to be affected by the independent variable. When conducting experiments, there are often many factors that could influence the outcome, and so researchers can't always feasibly eliminate all of them, but they do their best to control the unwanted variables by canceling out the effects using control groups. So a control group is a set of subjects that does not receive the specific factor being tested, but is otherwise identical to the experimental group. And all of the factors that are identical between the experimental group and the control group are known as constants or controlled variables, depending on which textbook you're using. Okay, so by having an experimental group and a control group, scientists can determine if, in fact, their independent variable has an effect on their dependent variable and decide whether the type or their hypothesis should be supported or rejected. And scientists often include two control groups. They're called a positive control and a negative control. A negative control is exposed to all of the variables an experimental group is exposed to, except the independent variable, and should give a negative result. So when you think control group, from what you've been taught in your previous science classes, you're probably thinking negative control group. But oftentimes a scientist is also going to use a positive control group. So positive controls use an agent that are known for sure to produce the expected predicted result, and they're very helpful in validating experimental procedure. So they let us know that human error didn't screw up the experiment. And they let us know that the reagents in the experiment are working properly. And not every experiment requires both control groups, but with either control group, if they don't work, that means that you can't be sure of any of your experimental results. Okay, so we want to take a moment and pause and practice. All right, so let's say that you're suffering from allergies and you don't really know what's causing them. So your doctor decides to do an intradermal allergy test. So what you can see in the picture is there's this white plastic piece with little pegs. And at the end of each peg is a teeny tiny needle, and each of those needles will have been dipped into a different allergen. And what they're going to look for is if you get a red, itchy, raised bump under the needle. That would be a positive test result. So different allergens might include things like pollen or pet dander or peanut oil or whatever, any of the things people could be allergic to. But one of those needles is always going to be dipped in pure water, and one of those needles is always going to be dipped in something that we know for sure will cause a reaction, like perhaps mosquito venom. So what I want you to do now is pause, and in a notebook, I want you to write down what's the independent variable, what's the dependent variable, what's the positive control, and what's the negative control. When you're done, hit play and we'll go over the answers. Okay, so I'm hoping you identified the independent variable, the thing being tested, as the allergens found on each of the needle points. The dependent variable is going to be the red itchy bumps. That's what they're going to measure at the end. Your positive control, the thing that should, no matter what, create a red itchy bump, is going to be that mosquito venom. And your negative control is going to be the water. So if we think about it, these two control groups are really important in this experiment, because if I poke you with 25 different allergens and nothing gets red and itchy, it could mean that you're not allergic to anything. But it also could mean that maybe the nurse or doctor administered the test wrong. Maybe they didn't push it deep enough in your skin. Or maybe the reagents, the allergens on the tip of each needle, were old, and so they're not producing the result like they should if they've expired. So your positive control is going to make sure that you do produce a result. Okay. Now, the alternative could be when you get poked, what if everything goes itchy? Now, one possibility would be that you're actually allergic to everything, right? But the negative control plays a role here. This is important because remember, it shouldn't produce a result. If it does, you might not be allergic to each of the allergens at the tip of the needle. You might just be allergic to the metal. If you know somebody that has like a cheap jewelry allergy or something, you know that there are allergies to metal, and that's a possibility too. So we need to know if that's the case. So this is a really good example of why positive and negative controls are so important. Also, I want to point out that in a controlled experiment, we say you only test one thing at a time, right? And so we could see we have a positive control, which is going to be our mosquito venom. We have our negative control, which we said is water. But then you could see that even if those two pegs are our controls, there's like 1, 2, 3, 4, like 8 more on there, right? And so what's happening is the doctor is basically running 10 different experiments at the same time on your arm, right? And comparing them all back to those control groups. So oftentimes in science, we run experiments that require a lot of prep work and they take a long time to get through. And so instead of running one experiment, we can run hundreds or even thousands of experiments at a time and generate a lot of data. All right. So now we want to talk about the difference between theories and hypotheses. In the everyday use of the term theory, we usually use it to mean an idea. So people will say something like, oh, it's just a theory. They mean it's just an idea, but that's not true. The word theory has a really different meaning in science. And it's easiest to understand if we look at it in terms of how it relates to a hypothesis. So first, a scientific theory is much broader in scope than a hypothesis. So for example, a hypothesis is going to be specific, you know, like I think my flashlight doesn't work because the batteries are dead. So if I replace the batteries, it'll work, right? That's like a very specific thing. The theory of evolution is this broad general statement explaining that organisms are able to adapt to their environments over long periods of time, many generations. Second, a theory is going to be general enough to spin off many new testable hypotheses. And third, whereas a hypothesis is something that needs to be tested, a theory has already been tested many times and has a large body of supporting evidence behind it. So we have a high confidence in our theories by the time they become a theory. In spite of evidence supporting widely accepted theories, we are always learning, and especially as technology advances, we learn more and more about the natural world. And so scientists, remember, we never say we prove anything. We are totally willing to replace or revise theories when new evidence dictates that we should do so. So a really good example of this is that scientists once lumped bacteria and archaea together in one kingdom called prokarya. But with DNA technology advancing, we have DNA evidence that actually shows archaea are more closely related to eukarya. So it's important to note that any truth that there is in science is always conditional and always based on the available evidence. All right, so once we run an experiment and we have all of this data collection and analysis, and it's this long, extensive process, the next step is to publish your findings. And so scientists often work for years, even like decades, trying to answer questions about the natural world. And once they get all that data together, they want to get it published as soon as they can. And the reason for that is that in science, generally, the first person to publish gets most of the credit. It allows researchers to share their ideas and to evaluate each other's work. And there are a lot of reputable scientific journals, including like Nature and Science. And they will only publish articles after they've gone through a rigorous process of peer review. Now, probably you've heard the word peer before, and you might know of your peers as like your friends, but make no mistake, peer review is not sending your hard work to your BFF and having them proofread it. So I want to take a step back and go through this process because it is intense and painstaking for a scientist. Okay, so to understand why it's important, the first thing you need to understand is that you're never the only person in the world working on answering a question. There are often people all over the world trying to get to the same answer. And like we already said, whoever publishes first is going to get all the credit. And so that creates intense competition between scientists and a race to be published. So if somebody else publishes your topic before you do, it's called getting scooped. And it's something that every scientist fears. Nobody wants to get scooped. Anyway, when you're ready to publish your article, you submit it to a journal. And if it shows enough promise, it's going to be sent to peer review. But like I said, it's not your best friend that gets to see all of your hard-earned work. It's going to be an anonymous, independent expert, somebody else in your field studying the same thing you are, and they're going to get all of that hard-earned knowledge, your years of research, your data, your controls, your data analysis, your conclusion, your experimental conditions, all of the details of what you've been doing. So basically, their job is to look for oversights, unfair influences, fraud, bias, mistakes in technique, or reasoning that would invalidate your paper, right? And so basically, you give all of your secrets away to your biggest competition, and it's their job to tear it apart. And then once they nitpick every single thing that they think is wrong with it, the journal sends your paper back to you, and you have to address all of the things that they said were problematic with your paper. So it often means months of further experimentation and data analysis and whatnot. And then you can resubmit, and then maybe the journal will publish your paper. So while peer review doesn't guarantee that a piece of work is correct, it does certify at least that the work has met the standards set by the scientific community. And it seems really risky to send all of that work to your biggest competition who are trying their hardest to scoop you. So why do we require it? Well, first and foremost, it keeps people honest and it prevents them from making outlandish claims. For better or worse, once research is published, it enters like a dynamic marketplace, and with social media and the internet, it can travel far and fast. And it can have a profound effect on people's health and even things like public policy. So peer review from respectable journals helps to make sure that only the most robust information that we have a high degree of confidence in makes it into that dynamic. Unfortunately, with, again, everything being so easy and accessible on the internet, there have cropped up for-profit predatory journals that do not require peer review. They'll publish anything, and they can do a really good job of making it look incredibly professional and legitimate, but what they end up publishing is what we call pseudoscience or fake science. So a really good example of this, actually my favorite example of this, is in 2017, a scientist was kind of fed up with the garbage science he saw coming out of these predatory journals. And so he created an absurd Star Wars themed spoof paper. It was all about midichlorians. If you are not a Star Wars fan, I'm sorry, you should probably go watch the movies. But if you don't know this, a midichlorian is a tiny microscopic particle in the blood that's responsible for the Force. So what he did is he basically copied a Wikipedia page about mitochondria, replaced the word mitochondria with midichlorians, and published it under the author name, I think it was like Lucas McGeorge or something. So, George Lucas. Anyway, so he submitted it to all of these journals, and 4 of them accepted and published it. So, obviously that's problematic, right? If you read just a few sentences into it, you would see that it's garbage. And yet we have the American Journal of Medical and Biological Research, the International Journal of Molecular Biology, the Austin Journal of Pharmacology, and the American Research Journal of Biosciences. They all sound like legitimate journals, and they all published this totally garbage article. So the moral of the story is that as consumers, we need to do our due diligence and be educated about our sources and where we're looking for information. I have a list of predatory journals. It's on my website, and you guys will be doing some deep dive into some of that as we move on. So that is everything I have for you today. If you want to find that list of predatory journals or see the fake midichlorian article that was published, you can visit my website at www.sevengablescience.com and like and subscribe to my YouTube channel, 7 Gable Science, to keep learning. Thanks, guys.